Okay, your love bug meiosis lab is detailed here. Your lab sheet is in the blue basket at the front and on my big desk. Um, the materials that you're going to need for this, I have altered a bit from the instructions that you'll already find that I posted already on Schoology. So you're going to need this packet. There are baskets in the front of the room and in the back of the room that have little strips of green or pink paper that are going to represent the chromosomes from mom or from dad. So you'll need 2 green and 2 pink. You also need scissors and glue sticks, which are already on your tables. So I'm looking here at number 2. This is a key that we will use quite often in genetics. And what a key gives you, it gives you the traits. In this case, we're looking at the antenna and the spots of a love bug. It gives you the alleles for a trait. Alleles are the 2 alternate versions of a trait. So for the antenna, there are curled or straight antennae. For the spots, heart-shaped or round antennae. No spots, sorry. Got carried away with antennae. But you also notice some letters here. So capitals represent dominance. Lowercase represents recessive. So A, capital A, curled antennae, dominate straight antennae. Heart-shaped spots dominate round spots. Okay, so the first thing that we're going to do is we're going to kind of lay out what we have. So lay out our initial gene combinations from the parents. Sorry. So we have our green ones here, and the green ones, both of them have, since they're sister chromatids and they're identical, have the capital A's and the capital H's. The pink ones, lowercase a's and lowercase h's. And if you notice, I've already numbered our sister chromatids as 1, 2, 3, and 4. Over to the left, what you're going to put are the characteristics that are present in the chromosomes from dad and from mom. Step 2, this is where you have some choices, and what you choose to do may be different from the person sitting beside of you. It's up to you. So we're going to have a crossing over event, and we're just going to have one. So you've got to choose which crossing over event are you going to have. Now we know crossing over is going to have to occur in those genes that are closer to the edge and farther away from the centromere of the chromosome. So our A's are up at the top and our H's are at the bottom. So I've given you an example of how you could do this crossing over event. So I could switch the A's with my sister chromatid number 1 and my sister chromatid number 3. So literally what I'm going to do is I'm going to use my scissors, and you better make sure before you commit to this, you're just going to lob off the end, just and then you're going to switch them. So just flip them. Do a little switcheroo, and then glue the new gene combinations together. So when you do this little switcheroo, then you're going to end up with a sister chromatid. It's got a little pink on it. The rest of it's green. This one's got a little green on it. The rest of it's pink. So you can do that with the A's on the top end, or, so it's not both, it's either or. You'll do the H's at the bottom. So the example I show here is we could switch the H from chromatid number 2 and chromatid number 4. So cut them, switch them, glue them down. So now you're going to have one that's green, 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 pink, 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 green. So it's up to you, the A's or the H's, not both. Then you have a set of questions here in step number 3. I gave you some little hintsies there, so I put homologous chromosomes, blah, homologous chromosomes, blah. You will find the answers to these in a word bank. It's really a phrase bank that I've included in the original instructions on Schoology. Step number 4, we're just going to drag down. Well, I can drag it down because I have them here together because I scanned them in, but you'll have to flip it over. But I'm going to take this information here. So once you get your switcheroos done, go back in and put your A's and H's, A's and H's, A's and H's. Drag those A's and H's down here. Drag them down here. And then over to the sides, I've left you a place there to put the genotypes of these new sister chromatid combinations and some of those that weren't messed with. So by gene combinations, we're talking about the A and the H that's on the chromatid. So you're just going to write 2 letters down here, whatever 2 letters are on the chromatid. Then step number 5. What happens in meiosis 2 after this? So we know what's going to happen. There's going to be a division. And then if you'll complete these statements, again, you will find these statements in the word bank that is on the original instruction sheet in Schoology. Step 6, illustrate the results of meiosis 2 below. So now I'm not concerned with the letters that are coming down here. I'm concerned with the phenotype, the physical expression of these genes. What will it look like? So this is where you're going to go back to your key and you're going to use your knowledge based upon the key. Capital A is going to be the curled antennae. Lowercase a, I'm looking for the word straight. If there's a capital H, I'm looking for the word heart. If there's a lowercase h, I'm looking for the word round. So instead of having the letters, which I would still put my letters right there, I would write the word that those letters represent beside of them. And then number 7, you've got a couple of things to answer here. You're just circling them. And then when you're finished, I would like for you to take your chromosome pairs and glue them on the back of the packet. Now there is a second page. It is extra credit. So you'll see a second page. It says extra credit. Actually doesn't say extra credit. I'm saying extra credit. It just says none of these things. It doesn't say due Friday or extra credit. But that second page, that's a bunch of analysis and reflection, that is extra credit. So you can turn in your packet with or without the extra credit. But either way, it's due on Friday.